Hello. First of all, I would like to thank the organizers for giving me the opportunity to give this talk to you and present this foot-and-mouth disease LL3B3D vaccine platform, which is a collaborative effort between USDA, Animal Research Services, and Zoetis. I'm here on behalf of Zoetis. I'm not the project leader for this project. This is led from the US, and in particular from my colleague, John Harham. I'm also representing him because he has not been able to travel to the EU. And we have also here colleagues from USDA, Luis Rodriguez and Elizabeth Rieder, that we will also be able to help me to provide you some responses to questions that you may have at the end of my presentation. We have already talked about that, not only me or other presenters, about the complexity of the disease. You all know that foot-and-mouth disease is one of the most, or if not the most, largely economically important disease of livestock. And the complexity of the disease comes from the fact that there are several pools, different serotypes in different pools, and also different strains in the different serotypes. Luckily, there are vaccines available for this disease, and which has a very big economic impact. But we need that each pool requires a tailored vaccine. They are produced from wild-type strains, so there is a need to produce in high-containment facilities. We have extensively talked about that already. But there is, and I want to highlight that here, the risk of release of the wild type from the manufacturing sites. And we have to deal also with a lot of regulations regarding how these manufacturing facilities has to be. They are not fully DIVA, or they are not fully DIVA. They are compatible with DIVA because manufacturers make the effort to try to eliminate non-structural proteins to get this DIVA compatibility, but it's not 100% DIVA because we have seen that after multiple vaccinations we can have a positive result in this around 3–5% for these non-structural proteins, and thus this not allows the fully DIVA characteristics, which is differentiating infected from vaccinated animals. So there is a need from an efficacy perspective. Also, I have to say that the current vaccines protect from clinical signs, but do not fully protect against fever, viremia, shedding, or persistence. So there is a clear need to improve the existing vaccines. Again, we have all seen that map several times, just to highlight the complexity of the disease, again, with the different 7 pools that are distributed in different areas all over the world, different strains, different serotypes. And that makes that foot-and-mouth disease countries have to be prepared for any serotype that can come from any other region, and that can be a natural incursion, but also intended incursion. So foot-and-mouth disease countries have to protect against that, and they do that by a stockpile of vaccines. But as I said before, it's very difficult to predict which serotype, which strain is going to come into a region, a free region. So there is also the need to have a flexible platform with a rapid response capability to address the fact that any strain can come and any serotype can come to a free area in any moment. And here is where our vaccine platform here? Okay, sorry. I went too far.
Got excited.
Sorry. Oh my God. Yeah. Thank you. Sorry about that. So here is where our vaccine platform can play a key role, and I'm here to present you the platform. This is, again, collaborative effort. I want to highlight that between Zoetis and animal research services from USDA. And the objective of this platform will be finally to develop an inactivated vaccine. It won't be a live vaccine, it will be an inactivated vaccine adjuvanted with a proprietary oil-based adjuvant. The key features of this platform are three. The first one is the deletion of the Rieder protein that gives a full attenuation, that makes it very safe. Another key feature is the possibility to construct capsid genes and that allow easy swap of these capsid sequences from any strain. The capsid is flanked by 2 unique restriction enzyme sites, so this allows the change of the specificity, let's say, of this construction to any serotype by just using simple molecular biology techniques. Another key feature is that we have 2 independent and stable markers, one on 3B and another one on 3D. These both genes are genetically modified, and that's why we have 100% fully DIVA capacity on this vaccine platform. As I mentioned before, the current vaccines, inactivated vaccines, have a remaining non-structural proteins present, although there are efforts to eliminate them that can provide some false positive around 3 or 5%. These characteristics of this platform makes it a very safe and easy production technology. The genetically modified organisms are completely virulent and non-transmissible, and we have shown that both in cattle and swine, and I will show you afterwards some experiments that have been conducted to demonstrate that. And the platform is based on a backbone, the A25 Cruzeiro strain. We know how to produce that using methods that are currently available for other inactivated vaccines, and we have good yields. So this is using the same manufacturing conditions that we are using for current inactivated vaccines. And from an efficacy perspective, our proprietary adjuvant increases vaccine potency well above traditional vaccines, and I will also show you some data to demonstrate that. This is a slide that summarizes the safety studies that we have performed with our vaccine platform. As you can see, we have used different constructions from different strains. You can see them. Oh, sorry. You can see them here. We have used different constructions against different strains. We have used different inoculation routes. You can see heel bulb and contact, intralingual, aerosol and contact. We have conducted these studies in 2 species, swine and cattle. We use swine because you know that it is the most susceptible species, so the results obtained here are relevant for that. And in all the cases, the results were that after the inoculation of our vaccine candidate without any adjuvant, we had no clinical disease, we have no viral shedding, we have no fever spike, no contact transmission, and very limited, if any, immune response. That means that it's not replicating in the target species. So this is the whole studies that we have conducted to demonstrate the safety of this vaccine platform has been very critical because we have obtained very recent select agent exclusion from the US. You know that in the US there is a list of agents that have specific requirements to be handled in specific conditions, and in fact they cannot be handled in US mainland. They have to be handled just on Plum Island. And so for us, this we have received a waiver for the Secretary of Agriculture, so we can now work with this vaccine platform in our facilities. And this is very recent because we obtained that in last April in this year. So here is a summary of the study designs that we have used to demonstrate the efficacy of this vaccine platform. We have used cattle, 6 months to 12 months steers. We vaccinate at day 0, and we challenge at 3 weeks after vaccination. So a single dose administration. And during this vaccination phase, we took serum samples to evaluate serological response and also cellular immune response, cellular-mediated immune response. And after challenge at different intervals, we also continue to take serum samples and to analyze the presence of virus, viremia, and also swabs. Of course, during the same period, we also perform clinical evaluations to see the clinical signs of the disease. And after the observation period, which was more than 3 weeks, we also evaluated persistence. And for that, we took probank samples. You know that they are oropharyngeal. The presence of the virus can be detected, where the presence of the virus can be detected. We conducted 2 efficacy studies, one adjuvant selection study and a PD50 study. Both studies were conducted according to OIE guidelines for the efficacy in testing. Here you have the results from the adjuvant selection study. We compared our results to a conventional type vaccine, and we use 2 different adjuvants, our proprietary adjuvants, and you can see in the serological response graph, that here we have the serological response from the animals inoculated with just a saline solution. We have the results from our adjuvant number 1, the results from the commercial vaccine, and the results for our adjuvant 2. So you can see here very clearly very strong immune response induced by, in part, this is part due to our proprietary adjuvant. You can see that we have more than 1.5 neutralizing antibody titers, which is widely accepted to be indicative of protection in even more than, even earlier than 1 week. So we can probably say here that our vaccine administered with 1 single dose and using our proprietary adjuvant is going to be able to induce protection earlier than 1 week. So we will have a very quick onset of immunity. And you can see that there is statistically significant differences between the response of our vaccine adjuvanted with adjuvant number 2 and the commercially type vaccine, and that the response is quite constant. Regarding, we also evaluated the cellular response, and you can also see here that our adjuvant 2 also elicits a strong cellular-mediated immune response if we compare that with commercial vaccines and of course, with non-vaccinated animals. During the challenge phase, we also evaluated the prevention of clinical signs. And on the left, you can see the graph where the temperatures are represented. The non-vaccinated animals, the animals injected with only a saline solution, have a fever peak. So there is an induction of the febrile response. While you can see this is a threshold to consider that animals do not have fever. With our adjuvant 2, you can see that all the animals were always below this threshold, so no febrile response induced not as the mean and not in the individual animals. From the clinical signs perspective, the typical ones of the disease, we can see here on the right that both the commercial vaccine and our vaccine adjuvanted with adjuvant number 2 fully prevent the appearance of clinical signs. So according to these results, we selected Adjuvant 2 to continue our studies. And the second study we did was a PD50 efficacy study, as I said before, according to OA recommendations. For that purpose, we vaccinated different groups of animals with different doses, and here we also evaluated different downstream processes, so different ways of producing our vaccine. Here we have a control group, and we use the full dose, 1/4 of a dose, 1/16 of a dose, with downstream process, so we call here number 1, and full dose, 1/4 of dose, and 16 dose, what we call a downstream process 2. Regarding the presence of viremia, you can see the peak of viremia induced in the non-vaccinated animals, the animals injected with a saline solution. While we have, you can see here that all the other groups, we have a full prevention of viremia. Only there was one animal in the 1/4 dose and the downstream process 1. This slow peak represents only one animal that was very close to the limit of detection of the technique, and where we performed virus isolation, and we were not able to isolate any live virus particle. This is from the viremia point of view. So we can say that the full dose prevented viremia in both cases in using 2 downstream processes. And regarding the clinical signs of persistence, you can see that the clinical signs were prevented in all the groups. There is only one animal here that we have shown that because this was in our study, but the clinical signs here were not related with foot and mouth disease. We put that as because these are the results that we obtained in the study, but it's not related to foot and mouth disease. So we can say that all the doses, the full dose, 1/4 and 1/16, fully prevent clinical signs. And also from persistence, we can say that the full dose for downstream process 1 and the full dose for downstream process 2 fully prevent persistence. Again, we have here a positive result from one animal for downstream process 2 that was very close to the limit of detection of the technique, and that and where we performed virus isolation, and again we were not able to isolate any live virus particle. So as a conclusion of our efficacy studies, our vaccine platform using Cruzeiro vaccine or Cruzeiro strain as a backbone prevented foot and mouth disease lesions in 1/16 dose. So that means that at least we have a very potent vaccine with at least 16 PD50 according to the OE recommendation. We can say that vaccines, the good results, that the high potent results that we obtain here are also due to our proprietary adjuvant. We can say that we have obtained high serum antibody titers compared with conventional vaccines, and they are statistically significant. We have a more robust CMI response compared to conventional vaccines. We have complete prevention of foot and mouth lesions, even with a 1/16 dose that corresponds only to 0.5 micrograms of antigen. And with the full dose, which is the 8 micrograms, we prevented persistent infection with both downstream processes that we were analyzing. We can also say that we have prevention of fever and prevention of viremia in downstream process 2. And a significant reduction of sheathing has also been observed, although we have not presented the data here. Also to highlight here the importance of this DIVA-compatible vaccine platform. As you know, and as I mentioned before, there is still some positive results with the current inactivated vaccines. And the epitopes that are present in the currently available commercially ELISAs. One is 3B, And our vaccine platform is genetically modified so that the epitope that is recognized by these commercially available ELISAs is not there. So this is not recognized. And we have analyzed that with 2 other current commercially available ELISAs, which is the Prionics 1 and the VRND. And you can see that the serological results from animals injected with PBS is below the limit of detection. The same for our vaccine. This is a full dose. And when we have a weak or positive control, of course, the response is positive. And when using the VRND ELISA, we can observe the same results. No detection of our vaccine, so no detection of antibodies against non-structural proteins in our vaccine. So as a conclusion for this vaccine platform, we can offer protection of livestock from the threat of natural or intended foot-and-mouth disease outbreaks with full DIVA compatibility. We can prevent foot-and-mouth lesions even after only single vaccination. We can say that this response is that we have a very quick onset of immunity. It prevents foot-and-mouth disease persistent infection. It allows differentiation of infected from vaccinated animals. It is safe in vitro and in vivo. There is no need to produce virulent foot-and-mouth disease virus, and you know that this is very important. And so this will allow us to have a global manufacturing capacity using our global manufacturing capacities, which is quite huge in Zoetis. No need to adapt or field strains to cell culture. So that means that this platform will allow a rapid response capability, and we will be very quickly able to switch from one strain for another. Finally, I would like to thank you again, the USDA, ARS, and in particular Elizabeth and Luis for their collaboration on that. And also I have to mention the FLI Institute in Germany, because we also conducted some safety studies there. So thank you also to Michael Schmauer. And just to let you know the people from Zoetis that is collaborating in this project, led by John Harham in the US. Thank you very much for your attention.